Hello, Year 9, Mrs. Corbett here. So we're going to be looking at microscopes today. Now, there is another lesson that goes with this that is about the required practical and how to make a slide. You will have the opportunity to do that, but I have made a video on that for you as well. So let's kick off with labelling an average-looking microscope that you might use in science. And there we go, there are the answers. So we have got A, the eyepiece lens over here. We have got B, which is the focusing wheel. And there are two of those. The bigger one is called the coarse focus, and the little one above it is called the fine focus. And then we've got the mirror, which could be replaced by a light, and that goes through the stage, which is E, and D is the objective lens, and F is the slide on top of the stage. Now, you need to know, to be a grade 5 or 6, which is the minimum you really should be aiming for, is look at the difference between magnification and resolution, describe the advantages and disadvantages of light and electron microscopes, and you need to be able to use the formula. If you can compare the different microscopes, even better, so giving similarities and differences and justify the use of one, then that's going to take you up to a grade 7 or 8. So here's a nice little starter. If we have an eyepiece magnification of 10, an objective magnification of 4, so the eyepiece lens is where we put our eyes, the bit we look through. The objective lens is what's looking down on your slide. Now, quite simply, you can pause it here and work these out, but you multiply the two together. So 10 times 4 is 40, etc. Here are the answers. So you might wish to have gone back and paused that. So let's look at the most basic microscope that you're likely to use in the lab, and that is a light microscope. Now, this little bit at the bottom is a mirror, but we also have ones in science that you most likely have used that have a light. So you would normally direct that mirror towards the light, so you get light reflecting back up the body tube and up the lenses so that you can see clearly. So at the end of this PowerPoint, there is a microscope slide, so you might wish to print that off and have a go at labelling yourself. Here is another one here. So just have a quick look. Can you remember, where is the eyepiece lens? Point at the screen. Where is the objective lens? Where is the coarse focus? Where is the stage? Etc. Okay, eyepiece lens times objective lens gives you the total magnification. Now, this is quite a basic one. There are also electron microscopes. You don't have to label those, but the two main types of electron microscope are the TEM, the transmission electron microscope, which passes electrons through the specimen and dispersed by the structures there. And there's also the scanning electron microscope, which coats the specimen in a thin layer of metal, and a beam of electron bounces off the surface to form a 3D image. The reason why people tend to use electron microscopes over light microscopes is that they have such a greater resolution. I'm just going to show you some pictures taken from electron microscope. From a light microscope, you will see structures, basic structures. But from an electron microscope, we can use, so at the top there, number 1 is chloroplast on a light microscope, but there we've got the other two microscope images. They are in much closer, much more depth. And as you can see, the SEM microscope is really, really fine detail. And here's another, the left-hand picture is with a light microscope, and then much, much greater resolution, much greater magnification used with the electron microscope. So this is extra detail. I'm not going to spend too long on these, but these are the two types of microscope and how they work. So the transmission electron microscope is similar to a light microscope, but it uses a beam of electrons instead of light, and it has a viewing screen at the bottom. They're much larger than the microscope. And then you've got the scanning microscope, which has a specimen covered with a thin layer of heavy metal, and so therefore it reflects off of the beams of electrons. And as you can see with this chap sitting next to the microscope, it's quite a lot larger than what we're used to and has to be displayed onto a video screen. What you do need to be able to look at and compare is, okay, if I had this specific thing that I needed to look at, an organism or an object, which microscope would I use? And so this is quite a good time to just double check in on that. So if I was trying to look at DNA, what kind of microscope would I use? If I was trying to look at bacteria, what kind of microscope would I use? What about a blood cell? What about an atom? So a light microscope uses the waves of light, so 400 nanometers, and the resolution is 200 nanometers, whereas with an electron microscope it's 0.5 nanometers. And look at the magnification. I mean, huge amount more magnification on an electron microscope. So if we're looking at size-wise, the blood cell here is going to be the largest, followed by the bacteria, followed by the DNA, followed by the atom. So we just about see a decent blood cell on a light microscope. But apart from that, you'd see a bacterial cell on a light microscope. But for real detail, high resolution, then we're going to need to use that electron microscope. Okay. So there we go. That's what we can see with the human eye. So a light microscope, we can see our bacteria, we can see our cell, but for an electron microscope, we get a much better image of a bacteria, a much better image of a virus, and things like DNA. Not quite reaching an atom size there on that one. This is an activity for in class. So if you wish to complete them, they are at the end of this PowerPoint. Okay, so now we're going to look at the equation that you need to learn. You don't need to know many for biology, but you do need to know this one. So this is magnification, which is a measurement of how many times bigger the object is that you've shown that the microscope can show. So we would do that by doing size of the image divided by the actual size of the object. So the size of the image, you are normally given one. I'll show you one shortly in a bit, and you would have to work it out. So I've got one on the next slide. So just remember, magnification is size of the image divided by the actual size of the object. So here we go. So the magnification of this bacterial cell is 5,000. Okay, so quite small, magnified, quite great length. And can you see it's got a scale across the bacteria? So what you have to do is each division of this scale is 1 millimeter. So we can work out the image size, and we've got the magnification, so we can then rearrange the equation to work out the actual size of this bacteria. Now I'm looking at this, I'm going to do a little bit of a count. So I've got 1, 2, 3. So what am I doing? I'm just finding the diameter of the bacterial cell. So 5 times 1,000. So the diameter 1, 2, 3, 4, 5. We're excluding this part because it's just the diameter of it, which is half the width. There we go. So 5,000 micrometers, which means that we can rearrange the equation. So it's the image size divided by magnification will give us the actual size of the object. So I can range that. So size divided by magnification is going to give me 1. 5,000 divided by 5,000. So that's a really easy one. So there's a magnification task coming up where you have to work out the size of the object, and that's at the end. So if you want to go through and look at the magnification of them, and I'm going to go back, was to evaluate the use of an electron microscope and a light microscope, giving one example where each is used. So you would get 1 mark if you wrote that an advantage of an electron microscope is it has higher resolution, 1 mark, higher magnification than a light microscope. It can see structures in more detail or organelles in more detail. If you just put more detail, that's not enough. You have to give an example of what more detail we have. Disadvantages of an electron microscope is they're rather large, they take up a huge amount of space, and they're very expensive, and can potentially have some dangers to them in that you're using high beam electrons. And if you stop those at an angle, you've got X-rays. A light microscope, an advantage of that is it gives clear structure. It's a small, useful microscope that allows you to see clearly the structure of small living organisms. Disadvantages is that it doesn't give clear resolution or magnification of non-living organisms such as viruses because they're far too small. And so we've given there an example of where each type would be used. So an electron microscope would be to see structures of organelles, parts of DNA, viruses and non-living organisms, whereas light microscopes are very much to give an outline of structure for larger organisms. So if you said any of that, give yourself some marks. And here we go, here's some more. So magnify up to 2 million times. They've got a resolving power of 10 nanometers. These are all really specific ones. Subcellular structures, more details of organelles, that's the same thing. Light microscopes can up to 2,000 times the resolving power, and they can look at cell sliding as well. So there's some more examples. So if you were able to compare and contrast, if you were able to get some really good evaluative points, then you're at that 7 to 8 grade. We didn't specifically compare, we evaluated, but if we were comparing, we'd look at similarities or differences. So they both enable us to magnify things and look at things with a more increased resolving power, etc. What else could you do to go further? Everything beyond this point of reporting. So we've got some units that we can work out, how big things are, and look at our units. So 1 millimetre is 10 to the minus 3 metres, or 0.001 metres. So if you know that magnification is an issue for you, then you're going to have to have a look at these and have a go. So this is a bacterial cell. Now, if you look at a bacterial cell on there, 10 to the minus 6, that falls straight in the middle of an optical or a light microscope and an electron microscope. So we would look at those in more detail with an electron microscope. Viruses, very, very small, so that's why they're hard to treat, hard to detect, and can only, what am I saying, protein synthesis occurs. Hemoglobin, where oxygen binds to your red blood atoms. And we've also got some activities here where you can calculate the size, of the diameter of each of these. So this is an onion cell, and we've got here a kidney cell. We've got lots of kidney cells here. So you would just try and have a look at the diameter of one of them. And we've got a bacterial cell, and we've got a nerve cell. So there's lots there, and there's a key for magnification. That's all. There will be some other things that you can do attached to this recording, attached in the folder.